I will open it up for a general discussion. I hope we have a lively half hour of discussion.
Well, I have lots of questions for Jimmy, so I don't know if, but I don't want to hog. You should let your people ask but if nobody has, I have a lot.
So maybe start. Are there other questions with our audience? Don't be shy, please.
I think we'll learn something from hearing Denise's questions too.
I had a quick question for you, Denise. That was a great talk. So my name's Mitchell. I'm at NC State. I do a lot of cardiovascular modeling, but we deal with lumped parameter models a lot of the time.
Hmm.
Always this question about how many compartments do you need, but also how many compartments can you have and still have uniquely identifiable parameters. So I wanted to ask, how do you address that problem when you're picking the number of compartments that are needed to address the problem that you're dealing with?
Yeah, that's such a good question. So I have two answers. One, we're actually working in parameter identifiability right now. There's a lot of work been done in ODEs. Marissa Eisenberg and Marissa Renardi from my group have a paper coming out on parameter identifiability in PDEs, but agent-based modeling, all bets are off. So now let me get back to the real heart of the matter. So the way I see it is that all models that you build are phenomenological and mechanistic. Sometimes you have a real mechanistic representation of certain things, and sometimes in the same model you have a very phenomenological, which is what you're saying is like lump a bunch of things into representing that term. Okay, so what I think happens is when you do your uncertainty and sensitivity analysis, it'll tell you, okay, this is important, this is important, and this is important. If it keeps coming up that the phenomenological representation is important, then my suggestion to you is it is time to build a submodel that actually blows that out and you do the detail. And then you're able to, in a very smart way, if you don't want that running all the time, shut it off and then replace it with very sleek term that really captures the main thing that you learn from that submodel, rather than just having like a rate of something coming in, but it's capturing it in a little bit wiser way. And that's informed by actually doing the full blown out submodel. But that's exactly what we do, what you just described. That's how we do it. Because, I mean, you have to make some choices of what's going to be in and what's going to be out. And so in the mesoscale in particular, you're very myopic in what's going in your petri dish, right? And then you decide, okay, well, I'm just going to ignore, that. But then later something tells you, gosh, I really have to pay attention to that. So.
Awesome. Thank you.
Mm-hmm.
I have a question, and I'm asking the same question to many people. So you use the term prediction, which is kind of problematic because we really cannot predict the future. And anyone who claims there is a big problem because they have to prove it, and usually they can't. So now my question is like, you also mentioned that you validate stuff. And here, this is a problem that actually was something that was addressed in our white paper that we're coming out from the integration working group. You do a lot of integration work. And my question is specifically, how do you validate all those small little components? And actually, how predictive are you? Are you really doing prediction? Can you actually predict something? Or are you more talking about forecast or projection?
Okay, good question, Jacob. And so what I think, actually, we're predicting backwards, not forwards. We're saying, why did that happen?
So you're not predicting, you're trying to explain.
Yes, there you go. Sure.
Thank you.
Yes.
Thank you. Important correction.
Yes. And then number two, your second part of your question was the validation piece. So we actually take our cues from the experimental world to validate their mouse model of something, human disease. What they'll do is do knockouts. They'll treat it with certain things and ask, do we see the same sorts of behaviors, dynamics over time as what's seen in actual situations? And so that's exactly what we run our models through. We run it through all the same knockouts, validations, these sorts of things. And those are some of the main ways that we validate our models to see that the behavior is overall working right. And I will also be very clear to say is any data that we use to build our models, we are agnostic to when we validate. That's a completely different set of data that we use to validate the models that we use to calibrate and build our models. And we put blinders on for both. So that way we don't jade ourselves into, putting something in and saying, oh, look what we got out. Well, of course you got that out because you put it in. So we don't do that. So we try to be honest about it, because we think that's really important, particularly to convince our biology colleagues that what we're doing is sensical.
Yeah, these are good answers. So first of all, it's like, it seems that we need to change the language a little bit. This is why I'm saying, because the term prediction is problematic, because people who see this outside academia, they say, oh, can they really predict? And the answer is typically no. We cannot still predict. We're very far from the ability to predict. But so this is a correction I'm raising this question again and again for a reason. It has to be corrected at our level, so that people who read the papers eventually believe that's like we can actually do something useful. We don't have to convince only the biologists. This is like at our level, we have to understand this. But more importantly, you said that you have different pieces of data. How many pieces of data you have, and how do they look like? Give us an example of something where you have a different sets, and tell us for the different levels of multiscale exactly what you're doing, because this is important for us to learn.
Okay, so I'll try to do this quickly. Let me tell you that what I think, first of all, we have a paper, and maybe there's somebody who's doing an amazing job of like finding papers and sticking them in the chat. I don't know who that is, but you're awesome, whoever you are. I love that idea. If you can look up the word CaliPro, CALIPRO, and find that paper. We just recently published a paper called CaliPro. I think it was in BMC System Biology. I cannot remember what journal. But that paper shows you that we use, when you have different types of data, is when you actually do. Let's see, I'm just looking. Yes, I have different types of data here. You need different types of approaches. So can I share the screen for a second? Is that all right with you? Thank you.
Go ahead.
Thank you, James. So for example, from the non-human primate data, we have flow cytometry data. We have something called Boolean gating and spice analysis data, which tells you different cells and what they're making. We have the number of bacteria per granuloma over time. We have all sorts of data. We also have spatial data. So this is something called a PET CT scan showing us which granulomas are, where they are, if they're hot, meaning that they've been incorporating FDG, which is a glucose marker, and we can track that. They can do this a few times on the monkeys before necropsy. And then we also get this immunohistochemistry data at the time of necropsy. So we have both spatial data as well as temporal data. And I think it takes a special approach to calibrate a model and validate a model to these different types of data sets. And so CaliPro is a method that we've come up with recently because we've thought a lot about this question, about what do you do with different types of data and multiple types of data that you're trying to simultaneously make everybody happy at the same time. So my short answer is you have to read that paper. And if somebody just found it and put it in the chat, thank you. Thank you for putting it in the chat.
Thank you.
So, hi, I actually have a lymph node question. If I'm coming through.
Please go ahead.
Both these talks were amazing, which is not a question. Do you think the lymph node pressure drops findings that you're seeing are also going to apply to non-professional lymph nodes, the lymphoid tissue in the nasal passages, in the lungs? Do you think your findings are going to be applicable there as well?
Yeah, so there are lots of sort of lymph node-like structures that get set up around the body, either permanently or temporarily. And the answer is I don't know. I would suspect not, because the lymph node is really sort of a little factory unto itself with its own blood vessel supply. And I think these tertiary lymphatic organs are a bit looser in structure. So I doubt it's going to be such a cut and dry case of, there are some definite afferent vessels and definite efferent vessels that we can use to do the measurement. For example, with a lymph node, and I'm not sure that's the case with these other lymphatic structures.
And then following up on that, and this may be incredibly naive, but do you think your findings are, for normal lymph nodes, are they lymph node universal? Are there differences otherwise in lymph nodes associated with different parts of the body who are going to have different expected routes of antigen delivery?
Yeah, I wish I knew. You know, this is that I should explain. Those were mouse lymph nodes
Okay.
that we were doing. The mouse lymph nodes are about 1 millimeter in diameter, so an order of magnitude smaller than a human lymph node, roughly. And as far as I know, that is the only measurement of lymph node flow resistance in the history of mankind.
Oh, it was amazing.
Well, it's a cool experiment for sure, but it's a good example of how little we know about the lymphatic system just because there are not that many people doing it. I wish we knew more.
Oh, I thought I didn't know because I had missed it in the literature.
Oh no, it's not there.
Thank you so much.
Thank you for your comments.
This is John Rice. I have a question. And by the way, all due respect to people who ask naive questions if they think they're naive and aren't afraid to ask them. Thank you. But both of you seem to have a pretty well integrated model and then laboratory type of research capability. And Denise mentioned something about using experiments to validate models. And I'm curious about the degree of experimental design associated with testing a model. And is it you have a laboratory experiment that tells you something and you build a model to test what the laboratory said, or you have a model and now you're going to do a laboratory experiment to test whether the model could conceivably be right? And so in the design of experiment, classical scientific design of experiment side, so you take the model and you develop a hypothesis, and then you test a real hypothesis and your hypothesis about how something works is, that if you manipulate a variable in the model, X happens. And so the experiment is that if I make that manipulation in variables in the real world, the same thing happens to some degree of probability.
Yeah, Jimmy, why don't you answer that? Because you do a little bit more of the experiments, and I think my answer is a little different than yours. So why don't you go?
Yeah, well, I think what you said earlier, Denise, was our approach is we try and build our models based on a certain set of data and train them and whatnot. But then when it comes to validation, we grab some set of data that the model's never heard of before. And I think that's a really crucial thing to do. I mean, if you teach your dog to bark whenever he sees the postman and the dog barks when he sees the postman, you can't really brag on that, So I think it's, we benefit a lot from the literature. No one could do all of the experiments that are necessary to explain such a complex system. But that said, we use microfluidic chambers. We try to get human lymphatic tissues whenever we can. Unfortunately, we have an almost infinite supply of human lymphatic tissues that we can get from ovarian cancer surgeries. We just try and get what we can because it's really difficult with the lymphatic system. You cannot measure pressure flow or diameter in any human lymphatic vessel to any degree of accuracy that would be useful for informing the kinds of modeling we do. So we just have to rely on whatever is out there.
Agreed.
I found your bypass experiment really interesting. We were working in liver, and what we found doing intravital imaging was that in fact a lot of the liver sinusoids are shut down a lot of the time, and that you actually see a lot of retrograde flow. You don't expect to see blood going the wrong way in capillaries, but it happens a lot in the liver. And so I suspect that probably just the way some of the sort of truisms that people thought about, maybe perhaps the way you had flow in the lymph nodes, were because people had looked. We saw that when we went and looked at blood flow in the liver. And so I thought it was really fascinating to your comment about the possibility of having antigens shut down a bypass pathway. There seems to be bypass pathway in the liver, as it happens, which is important in certain situations. And so there may be some, I mean, they're very different structurally, but there may be some analogies where you don't have active gating, you don't have pumps, but you still could potentially be getting these kinds of flow control through slightly more subtle or less obvious mechanisms. And that really could be, I found that fascinating to hear.
Yeah, thanks. The liver is another fascinating organ for sure, and is chock full of lymphatics as well. So it's yeah, it's interesting. I hadn't seen that paper. I'd like to read it.
Okay, I'm gonna ask my question.
John Yin. We've got multiple Johns, so go ahead, please.
Thanks. So this might be for both Jimmy and Denise. Perhaps overlapping Jimmy's interest in devices and Denise's on granulomas. I was wondering if either or both of you could comment on challenges and opportunities for microfluidic or 3D in vitro models of granulomas.
Go for it, Jimmy.
I was going to ask you how they're constructed, so I obviously wouldn't know how to build one, so you go ahead.
Well, there's actually a new push at the NIH for in vitro granulomas, because there's a lot of, cost. I mean, the monkeys cost $20,000 each plus $20,000 a year to house, and a little different than mice. So it's a very tough system. If it wasn't for Bill and Melinda Gates, NIH can't afford that 300 colony of monkeys. So they need alternative approaches to these, and so one of them is this in vitro granuloma system. But and believe it or not, there were mouse bead models, there's other systems, and there's been some microfluidic work done, but it's all in the very, very early stages. But I think it's a direction that NIH is pushing, because they want, it's kind of a system that sits between the modeling and the actual, it's like halfway in between. So I think we're going to start to see more data coming from that. The question is, how different is what we see? It is so different in the mouse than in the primate. It's hard to even imagine what's going to happen when you even remove the in vivo piece and make it in situ, or what you're suggesting there. So I'm not really sure how much. And there's been some data saying we're going to learn a lot from it, and other data saying it's not useful at all. So I think it's still out on what's going to happen with that.
Well, I'm very curious about that particular comment, the difference between the mouse and the primate.
Yeah.
Is there anything that one can build from a scaling perspective? Scaling of animal systems, so mouse, rat, primates, whales, I don't know. But so what are the key features that are different between the mouse I don't know if it's dynamics or if the size or...
it's not size. It's definitely, it's not allometry. If it was allometry, we would be okay, but it's not allometry. One of the main reasons is that the immune response is very different in mice than humans. So for example, in mice, only Th2 type cells make IL-10, but in humans, a lot of T cell types make IL-10. So just that change alone is, now there are some humanized mouse studies. There's something called the Kramnik mouse with a capital K by the Igor Kramnik. It came up with this model where he was able to make a model that made granulomas that look like human primate granulomas. But the issue is that mice don't even make granulomas. There's this infiltrate and stuff gets there, but it doesn't even form these beautiful structures, which defines TB, frankly. The granuloma is the definition. I mean, tubercles comes from tuberculosis, which are calcified granulomas. So there's something clearly different about the migration, and I don't think it's a size issue. And I'll tell you why. Because rabbits form glorious primate-like granulomas. Not much bigger than a rat or a mouse, but yet they're perfect. Why don't we use rabbit models? Because there are no immunological reagents for rabbit models. So you cannot use it as a scientific model for TB. Because no one's come up with, for the primate stuff, we use all human homologies. So we're using human antibodies, we're using human probes, but there's nothing like that for the rabbit. So no one has developed that technology. So that's why but we use it because we can excise these granulomas and treat them with drugs and take beautiful pictures, and so we're able to get some information, but no immunology from the rabbits.
So could you comment on any differences in structure and dynamics between non-human primates and humans then?
Very, very similar. In fact, size that's an allometric situation there for sure, with the humans and the non-human primates. And as I said, one has 5 lung draining lymph nodes, one has 7. So there are some differences, but in terms of the overall disease and outcomes and phenotypes that we see in disease, there's many, and the same bacteria, infects both. So that's important as well. Even though it doesn't happen naturally in wild, it happens in the lab. So yeah.
Great, thank you.
Yeah, sure.
Other questions? I see John and Ruchira have their hands up. Anybody else before we go back to John and Ruchira, since they've already asked a question or two? Veronica, were you going to ask a question?
Oh, sure. Hi, Denise.
Hi, Veronica. Nice to see you.
I just thought that I'm wondering if you look at this question, because there is another very common disease, sarcoidosis, that also have this granulomas.
Mm-hmm.
And is it like sarcoidosis is a light form of tuberculosis that is kind of appear and disappear in response? Because can you comment? Maybe you look at this.
Sure. So it's really interesting because this granuloma thing is pretty unique to TB. Okay. However, as you're suggesting, sarcoidosis caused by a sort of a yeast parasite is very different, and yet it forms it. The difference is the granuloma that forms in tuberculosis is what we think of as Th1 driven. It's driven by inflammation of multiple cellular mediated immune inflammatory things. That the sarcoidosis granuloma is actually a Th2 driven granuloma. It looks very different, and it's comprised of very different types of cells. And so that alone is so interesting. And I'll tell you something else. The lungs are actually thought to be very Th2 dominant as in its resting state. So in other words, you wouldn't want the lungs. Remember, you breathe in about 100,000 particles of crap all day long. Yeast, pollen, dust. I mean, all kinds of things all day. Viruses, things like that. We would be, if your lungs were sort of default Th1, you'd be like getting pulmonary infections like all day long. But for the most part, our macrophages take that stuff up, chew it up, spit it out, and we're done. But so it takes a lot to turn the whole system of the lungs to a Th1 response in response to TB. It's like taking a tank and turning it around, where the sarcoidosis thing is a little bit different because the lungs are already default Th2. So actually sarcoidosis granulomas form much quicker and differently than the granuloma ones. So similar idea, very different mechanisms of evolvement, but it's an open area. I wish people would do it because I think it would be really fun to sort of use our stuff and study use our ideas and study a different disease. So I don't know if that answered your question, Veronica.
Yeah, but do you know if actually sarcoidosis is more prevalent in the areas where tuberculosis is more prevalent, or
no?
There is no.
Sarcoidosis is different. There is some overlap for sure, but it's typically rural and farms and these kinds of situations where, and remember, TB, you have to be crowded and lots of repeat exposure to get it. So that's going to be more like not rural, more in cities, more, but everybody's taking the same bus to the same coal mine every day, that kind of a thing. That's pretty much the TB story.
Th1 and Th2 separation is actually very interesting.
Isn't that really cool? Yeah, and I will tell you that I read that about a decade ago when I learned about them. So I haven't followed that literature. So maybe I'll learn something, maybe that they've evolved that literature more. So if there's anything there, I'd love to hear about it.
Thank you.
Good luck.
I guess I'm going to take the privilege of being the MC and ask one more question on for Jimmy Moore, which is, I really was very intrigued by your discussion of adjuvants. And talking to Veronica earlier today, we were discussing one of the key things, of course, is the duration of immune response. Back a year and a half ago when this all mess started, I remember posting on Facebook, the only thing that matters about COVID is how long the immune response lasts. And you made the very interesting comment that depending on the presentation of a particular antigen from a vaccine or potentially from the infection itself, you could excite very different kinds and types and durations of immune response. Can you give us any more insight into what your thinking is about ways forward? What would be interesting things to research to try to use the kinds of insights you have to understand the duration of immune response? Why do the same B cells churn out antibodies in one case for months and in other cases for a lifetime, for example?
Yeah, these are great questions, and I wish I knew the answers. You know, I would guess that, you know, for something like COVID, where you know, maybe the antibody response is transient, perhaps, we don't know, but maybe the T cell receptor response is what you're after. Then I think that's where we would come in and say, all right, well, this is the right adjuvant you need to induce that kind of immune response. The lymph node would need this time course of antigen delivery. But we just don't have the information to do that. But I think that's the kind of thing we're heading for. So, you know, what kind of response do you want? Okay, we got it. We know, you know, based on this platform that we would build, what sort of delivery profile you might want to give, and therefore pick the adjuvant off the shelf or design one that does that for you.
So I just have a comment about that because, you know, I live a little bit in the vaccine world for TB, and everybody says one thing: if you're talking about vaccines and you don't use the word adjuvant, then you're not talking about vaccines. And I also want to tell you something interesting. The most antigenic thing known to humans is the cell wall of Mycobacterium tuberculosis, and that's what Freud's adjuvant is. And
Yep.
it's basically fish oil and MTB cell wall, right? And when they do that little skin test, that's what it is, right? They're putting basically Freud's adjuvant under your arm. But you posed the question, Jimmy, about like what's the trigger, and I really think, and we've thought about this for a while with adjuvants, that the point of the adjuvant is to trigger the innate immune response, because it's the innate immune response that triggers adaptive immunity. And so you get in there, so I agree with you, it doesn't trigger adaptive immunity at all. I think what it does is trigger the innate immune response, which says, aha, we better turn on the adaptive immune response. And so maybe it's an indirect thing. And so if there's somebody that do that really early modeling where you're getting complement, you're getting, you know, TLR engagement, those sorts of things, I think that might be a key for that. But I think it's going to require somebody to really think about that, you know. I also wanted to ask you a question. So in the modeling that we did, and this was mice study, so it would be relevant, with Mark Miller on lymph nodes, you know, it's very interesting because dendritic cells leave the site of infection and they go to the lymph nodes. And of course, they stimulate T cells, and T cells leave and go to, you know, through the efferent lymphatics, and they go back to the site of infection. Dendritic cells never leave. If we stay in the efferent lymphatics, and we never see dendritic cells ever leaving. And I wonder, how could it be that you could be selective who gets to go and who doesn't? And I get the whole lymphadenopathy thing is happening, but isn't that interesting? So I don't know what you think about that.
Yeah, well, you know, it could be that their job is just to deliver the information, and once they're done, they, you know, they probably just die. But you know, T cell, they you know, there's the S1P receptor on T cells that guides them out. I think there's also a role for the high endothelial venules in lymph nodes for T cell delivery into the bloodstream. I'm not sure that's been looked at as heavily as it should be, because I think that's probably another route that T cells use to get out.
But if there's flow, it's like I feel like the DCs are hanging on and the T cells are going by them. You know, I have this image in my head of, you know, if there's flow pushing you out, you know, how do you keep from going? I get it that the T cells upregulate, you know, vascularization, you know, endothelial vascular to get out, but I don't get how the other guys don't get washed out. But anyway, I find that has always been very intriguing to me, that the D cells the dendritic cells don't leave. So.
Yeah.
Also, I don't understand what flow resistance is. I mean, I get what it is. I know what it is, but I don't understand what it does in the lymph node or why it's there. I'm a little confused about that.
Yeah, so keep in mind each lymphatic vessel can only generate a little bit of pressure, and it's got to use that pressure to push flow all the way through the lymph node, which is like a big sponge in the way. So it's a big resistance to flow, and what we've shown is that under normal conditions, when if you're not responding to anything, most of the flow will go around the low resistance pathway just to keep the fluid moving and stop you from getting edema in normal conditions. And only 10% goes into where the B cells and the T cells are. So that's sort of, 10% needs to be smelled for, potentially bad things happening. But then, that gets tripled with the shutdown of the low resistance pathway.
I see.
Yeah.
And then my last thing is, Tim Padera and Lance Munn do work on lymphatic transport. And I'm wondering, what are they doing that's different than what you are? Is there overlap, or are you guys asking different questions? Because I saw them give a talk last year and, in the systems biology stuff, and I'm trying to figure out, there seems to be.
They do great work. It's, I think what we do is a bit more physiology oriented, and, what they've done, and Tim's paper that came out in Science in 2018 on the metastatic cells going into the high endothelial venules is just fascinating stuff. It's really, really cool stuff. So, they're certainly more concerned with cancer than we are, but yeah, we overlap a lot. We have beers at the lymphatic conferences and enjoy each other's work for sure.
Okay, thank you. It was an awesome talk.
Right. Sorry, I know we're out of time. I just wanted to add, I found a really cool paper recently on the differences between T cell responses in mice and men. And this paper that I put in the chat shows there are orders of magnitude differences in the mouse system versus the human system in CAR T cell therapy is what this one's about. But it's a really cool paper. Great modeling. So I put that in.
Well, I hate to cut the conversation off, but in principle, is there one last question before we break? It's very late in London, so we don't want to keep our speakers.
This is John. As a community of multiscale modelers that the working group is made up of, if we could have two things that would best serve the community in the next 2 years, and with the two presenters, the kind of work you're doing, what would be the two things that would be the most helpful to you guys, that, the barrier that has to be broken or whatever, to make what you're doing more useful sooner?
You want to answer first, Jimmy? Or you?
Yeah, got an answer ready. Go for it. I'll think.
Well, I think computing power, I have to beg, borrow, and steal computing power. And, we get stuff on Exceed and we get stuff on NERSC. And, our models take a lot to run. So for us, we would like unlimited access to simulation power. And then number two, I need more. The thing is, I can build my models, but I can't spend time making beautiful GUIs and dashboards that I could share and let people play with and use, because NIH is not giving me money for that. They're giving me money for, to answer a specific question, but I can't actually spend time to make everything look all pretty and nice. And so I can share my models and usable and, translatable and things like that. So obviously Jakob disagrees with me, so he can answer that question.
I'm going to take executive privilege and give Jimmy Moore the chance to discuss what he thinks the most important thing is, and we'll let him close out the meeting.
Well, that's a great honor. Thank you. I'm constantly frustrated by, we're all frustrated when our research proposals get turned down. I think the sort of lack of appreciation for systems biology, for the chemical engineering aspects of lymph node function, it's just not appreciated by a large swath of the biological community. I think that's the one thing I would wish for is better communication amongst the sciences and a better appreciation for what other points of view can bring to a problem so complex as what goes on inside a lymph node.
Okay, well, I
Bless
want
you.
I want to thank our speakers again. We definitely need you to come back and talk more and have more discussion. I appreciate so much people staying on and being willing to join in the discussion pit. And with that, I'm afraid that we have to close the meeting. We will just have to say that we'll have you back. Thank you again. This was wonderful discussion and it was wonderful talks. Really.
Thank you all. It was a lot of fun.
Bye,
Thank you.
everybody.
We'll look
Goodbye.
forward to receiving the chat list because there were lots of references popped up in there. Thank you.
Yeah, a lot of reading to do before next week.
Oh, buddy.
Bye.
Bye-bye.